Hi, I'm Kevin Flanagan, Director of Translational Science at Cofactor Genomics, and I'm excited for this opportunity to share the work we've been doing at Cofactor Genomics to bring precision medicine to cancer patients facing tough treatment decisions, and to give doctors treating those patients a new diagnostic tool to make informed decisions about immunotherapies. So specifically, I'll be describing some work we've done in head and neck squamous cell carcinoma, as well as some of the work we've done in non-small cell lung cancer in order to bridge that precision medicine gap for patients facing treatment decisions. So I want to begin with the problem. The immunotherapy is really a story of promise and despair. The promise is that immunotherapy, specifically, I'm referring to immune checkpoint inhibitors or ICI, it's clearly one of the major medical breakthroughs of the 21st century. It's revolutionized cancer care and often leads to durable responses and even cures in diseases that otherwise have very poor outcomes. Because of this, they've been approved across 16 cancers with constantly expanding labels and massive sales to go along with that. The despair or the problem of immunotherapy is that it only works in a minority of patients. Not knowing who will benefit prior to treatment creates suboptimal therapy selection, overtreatment, increased adverse events, and poorer outcomes. So there are existing diagnostics, in particular PD-L1 IHC, but they're really substandard. They're poor predictors of response, and this poor performance leads to more aggressive therapies, such as ICI therapy plus chemotherapy on top of that. So I want to explore this a little bit more by starting with a case study of a patient treated by one of our partner oncologists. This was a 63-year-old white male with metastatic squamous cell carcinoma of the tonsil, which is a type of HNSCC or head and neck squamous cell carcinoma. The oncologist was considering several treatment options, including ICI as a monotherapy, ICI plus platinum-based chemotherapy, or non-ICI-based regimens, including a potential clinical trial. The patient had a tumor biopsy, which came back HPV positive, which is generally good for prognosis, as well as a PD-L1 combined positive score of 1 to 19. So what does that mean? Well, I'll be coming back to PD-L1 scoring sort of throughout this talk, so I wanted to orient you to PD-L1 scoring and CPS in particular, or combined positive score. So this is a diagram that I adapted from the ASCO guidelines published in 2022 for treatment of patients with recurrent or metastatic head and neck squamous cell carcinoma, or RM-HNSCC. So all patients with CPS of greater than or equal to 1 are eligible for monotherapy ICI. But for the most part, patients are receiving ICI in combination with platinum and 5-FU chemotherapy. So for patients with CPS on the bottom here, less than 1, that's the default first-line treatment. There really aren't a lot of good options for these patients, and outcomes are very poor. So patients are treated aggressively, and also guidelines encourage physicians to look for clinical trial options. For patients that are considered positive for PD-L1 or CPS greater than or equal to 1, that's the vast majority of patients. But unfortunately, as you can see, they can receive ICI as a monotherapy or ICI in combination with chemotherapy. There's really not any clear guidance on which patients should get which. Even in this group, most patients don't respond to monotherapy ICI, so it's common for doctors to default to giving the more aggressive ICI plus chemotherapy. And I should mention here pembrolizumab, that's the most commonly used ICI in head and neck squamous cell carcinoma. So because CPS doesn't give a great guidance for treatment decisions, in particular when we're just looking at CPS greater than or equal to 1, doctors sometimes use a higher CPS threshold to identify good candidates for monotherapy, usually CPS greater than or equal to 20, as you can see here. So in practice, most doctors are rarely treating patients in the CPS 1 to 19 group with monotherapy, but use the more aggressive chemotherapy plus ICI. And even in this CPS greater than or equal to 20 group, many of those patients in practice do get chemotherapy added to their regimen. So unfortunately, PD-L1, the standard of care diagnostic in this field, is really not very predictive and providing very little guidance, in particular in the CPS 1 to 19 group of patients. So returning to our case study, the problem here is that despite the testing done for this patient, the best treatment path is still unclear. CPS of 1 to 19 doesn't provide clear guidance between the more aggressive ICI plus chemo or the less aggressive ICI as a single agent. And so this is an issue that has continued to plague not just head and neck cancer treatment decisions, but across many cancer indications. And in fact, this is something we've heard from physicians across the United States as we set out to develop a solution. So these physicians described how in the absence of accurate predictive results, they're defaulting to that more aggressive ICI plus chemo treatment option. And they want a test, they really need a test that will guide patient treatment paths. So the solution that we have developed at Cofactor is OncoPrism. OncoPrism is an RNA-based multi-analyte biomarker that predicts response to ICI in cancer patients. Our approach is using something called health expression models. These are RNA expression-based models of immune cells and immune cell states. So using Lexogen's QuantSeq library prep kit, we generate RNA-seq expression data for patients with known treatment outcomes. We use then health expression models to recast that multitude of RNA expression data into meaningful representations of immune status of tumor, of the tumor microenvironment. So the result is a multidimensional biomarker that's trained with machine learning to distinguish between responders and non-responders, all based on the RNA data from the patient sample. So to reiterate this problem and make it more clear, this slide illustrates the challenge doctors and patients are up against when making therapy decisions. So this is kind of an important slide to get the overall idea. So I want to orient you a little bit. The green here represents the small fraction of patients who respond to ICI. The red represents the majority of patients who unfortunately do not respond. So as it stands now, if you look at PD-L1, the standard of care diagnostic, if you look at the performance in these patients, calling those to the left of this line as responders and those to the right as not responders, sorry, to the right as predicted responders. I'm sorry, this is backwards. So the left as responders and the right as predicted non-responders, ignore what's on the slide there. There's quite a few patients in red there to the left that are predicted responders, even though they don't truly respond. So in a perfect biomarker, everything to the left of that line would be red. So we're far from ideal, and there's quite a bit of room for improvement, and that's where OncoPrism comes in. So our goal with OncoPrism was to maintain the high sensitivity of PD-L1. It is capturing most of those true responders, but improve on the specificity. So narrow in just on those green true responders and not predict as many non-responders as responders. So with OncoPrism, we're really focused on narrowing in on those who are most likely to respond. So to do this, we launched the PREDAPT trial to build and validate OncoPrism. PREDAPT, or Predicting Immunotherapy Efficacy from Analysis of Pretreatment Tumor Biopsies, is a multi-center observational study enrolling patients from across the country. I think for head and neck cancer, we have a mix of 17 academic and community healthcare sites. And I'm going to be focused on head and neck cancer for this talk, but PREDAPT ultimately aims to validate OncoPrism in 11 different cancer types. So diving into head and neck cancer and the data, much of the work I'll be discussing has been published in the Journal for Immunotherapy of Cancer. So I encourage you to check that out for some additional information. Here's the basic design of our study in head and neck. So we used at the top there 1,205 total samples to develop and validate the test so far. Of those, 790 were used from public data sets for feature selection, and then an additional 415 were enrolled as part of our trial. We had a number of excluded patients or failed QC samples, and then split the remaining samples here at the bottom into a training set, validation cohort 1 of comprised of patients receiving monotherapy, and a validation cohort 2 of patients receiving chemoimmunotherapy, so ICI plus chemo. I'm going to be focusing on the monotherapy ICI validation for the rest of this talk, although we did see largely similar results in both validation cohorts. So for head and neck, we designed the test to predict disease control. So we define sort of response as having disease control. Disease control is any patient with complete response, partial response, or stable disease after treatment. So progressive disease patients, of course, do not have disease control. Our decision to design the test this way is validated by the data here. So we're looking at a Kaplan-Meier survival curve for progression-free survival. And we'll be looking at a few of these throughout the talk. So I just want to spend a moment just to describe the plot and make sure it's clear. So on the x-axis, we're looking at time in months from treatment to progression. So when does the cancer progress or the patient is deceased? The y-axis is the progression-free survival probability. So you can think of it as basically the fraction of patients that are still have not progressed along as we go along time. So in gray are the complete responders, in orange are the partial responders, and in black we see the stable disease. Then in blue, you can see the progressive disease. So not surprisingly, the complete responders in gray are doing great. The blue progressive disease are not doing well. They're progressing pretty much right away. But you can see here the partial responders in orange and the stable disease in black are really clustered together. And there's not a lot of daylight between them. So that sort of justifies grouping them together and including them both in the disease control positive class. And in fact, we saw a median progression-free survival, or PFS, of over 6 months for those stable disease patients, which represents a meaningful clinical benefit, especially considering that these patients otherwise have overall survivals often less than a year. Okay, so here I want to dig in a bit to the validation results. So we're looking at all of the monotherapy validation cohort patients lined up on the x-axis. Each bar then is a patient, and then the y-axis is the OncoPrism score, which is the output from the OncoPrism test. So OncoPrism scores range from 0 to 100 and are designed to be correlated with the likelihood of disease control. So patients are ranked by their OncoPrism scores and then colored based on their outcomes. So going from left to right along the x-axis, you have the lowest scoring patients to the highest scoring patients. And then orange patients had disease control. Gray patients progressed, did not have disease control. You can see that as you move to the right towards the higher scores, orange starts to predominate, and it's clear that the test is offering predictive value. Now, while the test generates a score from 0 to 100, in order to translate it into a usable clinical test in the clinic, the test actually doesn't report the score, but an OncoPrism group. Basically, these groups of patients with low scores are grouped together. Then the medium scores are grouped together, and the high scoring patients are grouped together into OncoPrism low, medium, and high groups. So importantly, the thresholds for these groups were set and locked using the training data, not this validation data. The training data set the thresholds, and then those are applied to this validation data and all additional clinical cases processed. So the goal then is that the low, medium, and high groups will translate into a pattern of increasing DCR in these groups. So here in the middle, that's what we're seeing. The x-axis is the OncoPrism group, low, medium, high, and the y-axis is the disease control rate, so the fraction of total patients in each group that has disease control. So higher is better. Higher means that most of the patients are benefiting from the ICI monotherapy. And you can see here that the DCR does in fact increase from low to medium to high. And that trend is significant by Cochrane Armitage test for trends. This black line shows the DCR for the overall cohort. And one thing I forgot to mention, it's important, is that the high group is actually the largest group. So nearly 50% of patients fall into that high group, really meaning that there's a large number of patients that can benefit and have strong predictive power from this test. So like DCR, OncoPrism groups are also correlated with progression-free survival. So just like the figure I showed you on the last slide, here we're looking instead at the progression-free survival for each OncoPrism group, low in blue, medium in gray, and high in orange. And as you can see, there's a significant trend towards longer PFS or progression-free survival as you go from low to medium to high. So this is important. The test works and it predicts DCR as well as PFS in response to ICI. But how does it compare to the standard of care, the test that's currently used, PD-L1? So we compared OncoPrism to PD-L1 CPS, as well as TMB, which is another biomarker that is sometimes used in head and neck cancer, but it's not widely used. It's not very predictive in head and neck cancer, so it's not used much. So looking at ROC curves is a good way to be able to compare performance across all the possible thresholds of a test. So that's what we're doing here on the left. Essentially, you look at the area under the curve, gray for PD-L1, orange for OncoPrism, and the higher the area under the curve, or the AUC, the better the overall performance of the test. So the red dotted line, that'd be a test that performs as well as chance. And then in gray is PD-L1 with an AUC of 0.62, which is predictive but not great. And then in orange is OncoPrism with an AUC of 0.73. So OncoPrism is beating PD-L1 there, and actually that's a pretty meaningful difference in AUC. As a reminder, our goal was to match the sensitivity of PD-L1, but improve upon the specificity. So did we do that? Looking in the middle here, using CPS greater than or equal to 1 as the cutoff, OncoPrism and PD-L1 CPS have identical in orange and gray there, identical sensitivities. But if you look over to the right, the specificities, OncoPrism is actually has 3 times higher specificity than PD-L1. Now, you might, though, then be distracted by this great big blue bar that represents TMB. So indeed, TMB in this population, this study, had very high specificity. However, it only identified a fraction of the patients with disease control. It was just 3 total patients. So its sensitivity was terrible, not really good enough for wide clinical use. And OncoPrism had 4 times higher sensitivity than TMB. So OncoPrism is really finding that sweet spot between these tests where it has high sensitivity like PD-L1 with 3 times the specificity, and that will translate well into the clinic. So circling back to this slide again, we achieved the goal of matching the sensitivity of PD-L1 while improving upon the specificity. And again, I apologize, those are predicted responders there to the left, and OncoPrism is really narrowing in on those patients without nearly as many false positives as PD-L1. It is important to note, though, that we don't intend OncoPrism to replace PD-L1. Rather, these tests can complement each other. So indeed, looking at 2 commonly used PD-L1 CPS thresholds, OncoPrism is predictive of DCR, and shown here PFS in CPS subgroups of greater than or equal to 1 on the left and greater than or equal to 20 on the right. So just like I showed you before for the overall population, from the OncoPrism group is correlated with PFS in these commonly used PD-L1 subgroups. So it's predictive even if you're looking just at those subgroups, and is able to inform treatment even on top of and in the context of PD-L1 results. So that takes us back then to the case study that I started with at the beginning of this talk. So this was the patient with a CPS in the 1 to 19 range, which did not provide clear guidance for a treatment path. So, and unfortunately, this is not uncommon. This is the CPS group of 1 to 19 represents about half or even more than half of all HNSCC patients. So this is a very common problem. Fortunately, the oncologist in this case ordered OncoPrism for this patient. and it returned a result of high. So this patient was prescribed monotherapy ICI and had a positive response to that treatment. So taking into account the OncoPrism results, the patient received the right treatment for them and was spared unnecessary chemotherapy that they likely otherwise would have endured, and avoided the risk of serious adverse events that goes along with that. So based on some of the results I've shown, OncoPrism HNSCC, we're excited to say, was granted Medicare reimbursement approval late last year, and we're in the process of scaling up the availability of this test to patients and doctors across the country so that there can be more success stories like in this case study. And building on that success that we've seen in head and neck cancer, we're expanding the test to additional indications as well. So first up is non-small cell lung cancer. OncoPrism NSCLC is built using the same underlying biomarker as HNSCC, but we use data from 1,454 NSCLC patient samples to understand what the distribution of patients looks like in lung cancer, and how it relates to their likelihood of response. So this guided us in setting up NSCLC specific thresholds for the test. And then we performed an initial validation using 51 patients from our PREDAP trial, with more continuing to enroll. So OncoPrism NSCLC predicts overall response, the more relevant metric in lung cancer, to ICI in stage 4 NSCLC patients. It's important to point out in NSCLC, unlike in head and neck cancer, the vast majority of patients are receiving ICI plus chemotherapy, even more so than in head and neck cancer. There really just isn't a great way to find patients that should get monotherapy, but OncoPrism can do that. So looking at the validation data, we compared the overall response rate, or ORR, for each of the OncoPrism groups in our initial 51 patient NSCLC validation cohort that came from 12 US healthcare systems. Looking at OncoPrism groups low, medium, high from left to right here, we see a nice stair-step pattern as you move up, indicating that there is a significant trend between OncoPrism groups and higher overall response rates. So exactly what we wanted to see, just like we saw in head and neck cancer, this test is predicting response to ICI. So comparing that performance to PD-L1, looking at the ROC curves, you can see that OncoPrism is, again, outperforming PD-L1. So as you can see, OncoPrism in orange has an AUC of 0.73 compared to just 0.60 for PD-L1. In particular, you see the separation at the early part of the graph, and that's indicative of OncoPrism's lower false positive rate, which is important in non-small cell lung cancer, where most patients are getting ICI plus chemo. It's important to have a low false positive rate in order to move them towards a monotherapy setting. So digging in a little bit more, here I'm showing you again on the left the OncoPrism overall response rate by group, and then for comparison on the right is the PD-L1 results. So here we're looking at PD-L1 TPS, which is tumor proportion score, similar to CPS that I discussed in head and neck cancer, but now it's we're looking just at the tumor cells that are positive, and that's the commonly used biomarker in non-small cell lung cancer. And using the common TPS thresholds of less than 1, 1 to 49, and greater than 50, greater than or equal to 50, which is used in lung cancer. So you can see there's no trend towards higher ORR with higher PD-L1. You know, there's a hint of higher ORR in the medium and high groups, the 1 to 49 and the 50 and above, but it's not a clear pattern, and it's not a significant trend. So all of this translates into better performance metrics for OncoPrism across the board. Here we're looking at accuracy, positive predictive value, negative predictive value, sensitivity, and specificity from left to right there on the graph. In each case, PD-L1 in gray has lower performance than OncoPrism in orange. Because of the design of the test and how we set it up and our goals, we're anticipating higher specificity for OncoPrism. But we were very pleased to see that we're also seeing higher sensitivity. And this will translate into more clinical utility for the test. So there's much more to come. We're continuing further validations in head and neck cancer, as well as, of course, non-small cell lung cancer, looking to bring these tests widely to the clinic. And in addition, we recently launched research use only versions of the test for colorectal, breast, bladder, and kidney cancer. We're continuing to validate and develop these tests further and hope to bring them to the clinic soon. And as I mentioned earlier, our PREDAPT study is designed to look at a total of 11 different cancers. So those are all sort of working their way through the pipeline, and we're very excited about that. Before I end, I wanted to highlight something that I think gets overlooked a lot in clinical testing, and that's the analytical performance of the test. So the success of the clinical validations I just showed relies upon robust analytical performance. Results need to be repeatable, reproducible, and robust against perturbation from contaminants or changes in the lab. So here I'm showing you repeated technical replicates of clinical FFPE samples. So these are samples with a wide range of quality and scores. Some of them, the RNA had very low DV200s. We report on samples as low as 23 DV200 of 23%. And on the top, we're looking at scores. On the y-axis is the scores, and then on the x-axis is a bunch of replicates of different biological samples. And you can see there's very tight performance, and these are all samples that were processed within the same batch as each other. So the replicates don't vary much. On the bottom here, we're turning it up a notch and processing samples across 8 different batches using different equipment on different days, different operators. But again, we're seeing very consistent performance. This reliability speaks to the robustness of the test and is really, again, it's instrumental in allowing the clinical performance to work. It is very important for clinical tests to be analytically valid and very low variability, high repeatability, et cetera. So this is continuing on that thought, and this is really getting into my acknowledgments in a sense here. OncoPrism relies on highly reproducible RNA-seq libraries. So as I mentioned, we use Lexogen's QuantSeq library prep. And stepping back from the scores, looking at the actual counts per million, the CPMs on the left here, the Spearman correlations between in this case, just one replicate, one biological sample replicated across multiple days by multiple operators. We see really high correlations. The scale here goes from 0.825 up to 1, so there's really high Spearman correlations, and that performance that we're seeing from our library preps and our RNA sequencing is critical to the performance of the test overall. There's more information about our analytical performance in this publication shown here, published in BMC Cancer. So I wanted to acknowledge a few other people. First of all, definitely the PREDAPT patients and their families that were really, obviously there's none of this. This is for the patients, and it's relies on the patients participating in our study, and we thank them so much for doing that. I want to thank our partners for Lexogen and this great opportunity to share with you the results of our work, as well as, of course, the entire team at Cofactor, and finally, all of our clinical partners. So we have involved in our PREDAPT trial and otherwise partnering with us. We have many cancer centers across the United States that have partnered with us and really make all of this work possible. So thank you to all of them, and thank you to you all for this opportunity to speak and hearing about our work at Cofactor Genomics.
First of all, I would like to thank Kevin for this exciting talk and the team at Cofactor for all the work they are doing to improve treatment options for cancer patients. Before we conclude the sessions, I would like to briefly introduce Lexogen and QuantSeq, the RNA-seq library prep technology behind OncoPrism. My name is Yvonne Güppel, and I'm head of product management at Lexogen. Lexogen is a biotech company founded in 2007 in Vienna, Austria, with Lexogen Inc. operating out of New Hampshire since 2014. Our aim is to bring innovation to the scientific community, fostering research progress, and ultimately improving patient outcomes with partners like Cofactor Genomics. Lexogen is ISO 9001 and 13485 certified to enable our partners to develop testing procedures using our products. We provide products, as already mentioned, for example, in form of kits for you to use in the lab, with a focus on high-quality, cost-efficient RNA-seq solution. Our services provide customizable NGS workflows, including various RNA-seq solutions, such as expression profiling, drug screening, whole transcriptome analysis, small RNA-seq, and biomarker discovery, but also DNA sequencing, sequencing only, and bioinformatics services. We also partner with other labs and companies to build new technologies and make NGS workflows more accessible for everyone. The Lexogen product portfolio covers the entire RNA-seq workflow, from RNA extraction and various pre-processing methods to different library prep solutions paired with data analysis, alongside with our 12 nucleotide UDIs for barcoding. Further, we offer different types of NGS controls and have application-specific end-to-end solutions, including workflows for difficult samples such as FFPE material, whole blood, extracellular vesicles, microbiome studies, or even plants. Our Lexogen NGS services are highly customizable and go even beyond our product offering. We routinely process bioinformatics requests and accept single-cell RNA sequencing, DNA-seq, and custom projects. We are most successful in challenging RNA-seq projects, can operate at scale, and have a proven track record for drug discovery projects. For today, I'd like to focus on Quantic, our highly robust expression profiling kit that builds the basis for Cofactor's OncoPrism test that Kevin introduced to you. Quantic is a 3' mRNA-seq library prep kit that pioneered gene expression analysis with its release more than a decade ago, and is since an established technology aiding cancer research in labs around the globe. Quantic provides an extremely fast and streamlined workflow in only 5 steps, thus not only saving time and consumables, but also improving consistency by removing particularly tricky steps, such as entry pair and adapter ligation. Quantic does not require any preprocessing steps, such as mRNA enrichment or rRNA depletion, and does not use RNA fragmentation. Instead, cDNA first strands are directly generated from total RNA inputs using oligo-DT priming. Following template RNA removal, second strand synthesis is initiated by random priming, generating adapter-tagged double-stranded cDNA. After a quick cleanup step, the cDNA is then amplified during PCR, unique dual indices and flow cell adapters are added, and the final cleanup step already results in the ready-to-sequence library. The whole workflow is completed in only 4 to 4.5 hours. While whole transcriptome sequencing covers the complete transcript and provides a uniform and even coverage, 3' mRNA-seq libraries such as Quantic focus reads on the 3' UTR and the ultimate exon. Quantic generates 1 fragment per transcript, enabling straightforward and highly accurate expression quantification by gene counting. In contrast to whole transcriptome sequencing, which requires assembly of the randomly generated fragments across the transcript body and normalization, the analysis of 3' seq libraries by read counting requires less computational resources, and results can be obtained much faster. As already highlighted by Kevin during his talk, the success of clinical validation strongly relies on robust analytical performance, ensuring that the results are reproducible, repeatable, and robust against perturbations. Due to its consistent within-batch and across-batch performance, Quantic is also suitable for highly challenging samples, such as FFPE material, which is often used in cancer research and is used to preserve and store patient tumor biopsies. It is also the material that is tested with OncoPrism. Corroborating the results shown by Kevin, our internal validations show a high overlap of detected genes independent of the sample preservation, comparing fresh frozen samples with a DV200 of approximately 90% and FFPE samples with a DV200 of only 9% at different thresholds. This is shown at the bar plot on the left side. In addition, on the right, gene count correlations between replicates for FFPE-preserved kidney tumor and normal tissue at dV200s of 45 to 50% show a very high correlation between technical replicates at only 20 nanograms total RNA input, similar to the results presented earlier by Kevin. The Quantsec technology comes in many flavors, from Quantsec Forward, which is highlighted here, to a pooled version for massive multiplexing, dedicated FFPE kits, or Quantsec Flex, which enables targeted sequencing, for example, in perturbation screens. Quantsec Ref is ideal for alternative polyannulation site studies. The first base of read 1 for Quantsec Reverse corresponds to the transcript end site. There are also custom versions available, as well as add-ons to effectively block globins or other abundant transcripts, such as BC1 and rodent brain samples. All of these options are also available as service. Due to its flexibility, Quantsec is used by basic researchers, as well as in applied and pharmaceutical settings, or for clinical research, with applications ranging from expression profiling or screening to high-throughput RNA kinetics in combination with LAMP-seq. Quantsec is therefore a fast, scalable, and cost-efficient solution for expression profiling. It's a very streamlined 3' mRNA sequencing technology. Pooled versions provide unparalleled scalability for multiplexing and enable processing of high-throughput projects while retaining robust and consistent performance. Quantsec is ideal for low input and low quality samples, and the method of choice for mRNA sequencing from FFP samples or gene expression studies from whole blood. Quantsec is a technology that is trusted by researchers worldwide and aiding to drive drug discovery, precision medicine, and cancer immunotherapy forward. These are just a few examples of publications using Quantsec. But if you are more interested in learning about OncoPrism tests, the use of Quantsec therein, or to read about the analytical performance, the multicenter validation for the predictive study, or the development of the biomarker classifier, check the select publications from our partners at Cofactor Genomics. With this, I would like to thank you all very much for your attention throughout the session. And if you are interested in testing Quantsec, get in touch with us by simply scanning the QR code, and feel free to follow Lexogen on social media for news, blogs, and other updates. Thank you very much.